Anjali Venkateshwaran, Senior Director of Marketing at Biomere, and it is my pleasure to introduce Tom Schofield, who will be joining me today. Tom is the founder and CEO of Splice Histology, that is a startup offering preclinical and clinical histology services. Biomere has recently partnered with Splice to offer histology and IHC analysis on samples dosed with novel therapeutics. Prior to founding Splice, Tom has over 20 years of experience in the anatomic pathology and histology space and has had positions at Quest Diagnostics, Beth Israel Lahey Health, and LabCorp. So, Joeen Biomere is a preclinical CRO with locations in Worcester, Mass., Richmond, California, and multiple sites in China. Our portfolio includes comprehensive PK and PD and toxicology services. And you can learn more about Biomere's service offerings at www.biomere.com. So, without further delay, let's get into today's topic on this really exciting partnership focused on preclinical histology services. So, welcome, Tom. So glad you could join me today. Let's start with an overview of your journey to founding Splice, because you've really built a long track record in this space. So what were the driving forces to start your own company?
Hi, Anjali. Thanks for having me. Great to be here. The best way to explain why Splice started was prior to Splice, I was a director of pathology for a large international company. And we saw a trend coming out of the pandemic where there was a severe lack of histotechnologists. And you didn't see the problem as much during the pandemic because there weren't as many biopsies to process. And then about in March of 2023, the COVID testing went away in a matter of weeks. And then my phone started to ring from hospitals across the country saying, I don't have enough histotechs. Can I send my tissue blocks to you and simply cut them and make a beautiful H&E and return the slides and the blocks back? To the point of 10,000 a day, we were turning away. So when you look into a little bit deeper, it's one of the few laboratories that's still manual. There's not a whole lot of automation. There's so many factors that go into making a beautiful histology slide. And this number of schools that have a program continue to, and there's, I think, about 26 left across the entire country.
Wow, really?
There's less.
Why is that? Why is that?
I think it's a lack of awareness of this profession. The average age was in the mid to upper 50s. I spoke at a conference last year, just explaining what Splice does, because in addition to doing histology, we also have a histotech training program. But to bring awareness, I asked the audience of 90 people to raise their hand if you're under the age of 30, and one person raised their hand. And I said,
oh my, yeah,
that's the problem. So, I spend a good amount of time talking with local schools, even high schools, about sharing the message of what is histotechnology. And certainly when I talk about the compensation, it's for an associate's level degree, it's about $10 more. So it pays pretty well. And I think folks that want to be play an important role in healthcare, but not deal with patients, this is a perfect profession. You're behind the scenes, but you're playing a critical role. So because we were turning away so many blocks per day, I said, we need to do something about this problem. We need to build an operation that can support places that need this service. And some hospitals were 4 to 6 weeks behind. I mean, these are all patients waiting for an answer. And internally, we need the right systems to manage our quality and document quality and share QC. It's all about quality for us.
Because as just to be clear, because in that regard, that's the data that's being used to make patient treatment decisions. So it's very, very critical that the quality is basically consistent and top notch.
Even your basic stuff, like, am I putting the right piece of tissue onto the right side? There needs to be internal mechanisms and systems in place to ensure that happens. So the company I worked at didn't really want to build capacity. So I had stepped away and started Splice at the very end of 2023. We're located in Worcester, Mass, right down the street from your offices. And yeah, I mean, we began work about 6 months later. Here we are. We're about 2 years old, and we have about 150 hospitals that have access to use us. And it's really interesting, like kind of why we started the company and where we are today.
Right.
Because the biggest thing was the problem isn't histotechnologists in the hospitals. The problem is the field of histotechnology, and that we've learned is expanded way beyond clinical. It's obviously
Right.
here we are doing research,
Right.
clinical
Right.
trials, drug development.
Right. And it's interesting because, I was just thinking as you were talking about like the histotechs, this is probably one of those AI, somewhat AI proofed professions, right? Because in my mind, when I think of histotechnology, I view it as an art. It is a science, but there is an art. And, there's a lot of human judgment that goes because, whether you're looking at, you're doing some gross, you're doing some grossing, even fixing, embedding these tissues. Some tissues are a little more robust than others. So I do think there's such a huge art component to it, which makes this a pretty, AI proof profession, something for people to think about.
You're absolutely right. Proper fixation, the fixation time,
Yes.
tissue processing, the orientation during embedding. Uh,
Yes.
those are all things that are just they vary based on the type of samples that you're working on. The humidity in the room,
Yes.
any sort of wind tunnels or vents will affect the tissue ribbon. So there's a
Right.
lot of variables. And while there are companies that have tried to automate it, I think we're still 5 or 10 years away from getting something that could reproduce what a human could.
Right. And even then, I would hazard a guess that yes, maybe some stuff that's more repeat, like a more robust tissue could be automated, but I would hesitate with when you have really soft tissues or something like the eye. From what the little bit I know about ocular, the eye is a tricky one. There's some hard tissue, there's some soft tissue, there's gel, there's matrices, and so I do again believe that you know I've been in in the business of, in the space for like over 2 decades. And for me, histopath analysis is so informative. You can actually see, okay, what's happening here? Am I getting tissue architecture changes? Am I seeing, damage? Am I seeing, and when you see it, it just speaks so much louder than, a number. And there's place for numbers, but I do believe that with histotech, high quality histotech data is priceless.
I can give you an example. We never intended to get into research until
Yeah.
we started getting calls from pharma and
Hmm.
development. I actually worked with a company that had purchased the blocks from a failed clinical trial. It was about 1,000 blocks, and it was
Yeah.
a celiac disease. And they sent it to us, and we determined that about a third of those cases were not embedded in the right orientation.
Oh dear.
They should have excluded a significant a portion of
Right.
those patients from the trial, which
Right.
ultimately would really dramatically impact. So you're absolutely right. There's a
Wow.
um, and on the
That
clinical
that that
side,
shakes me. That shakes me because yeah, that shakes me because this is something that's entirely avoidable, right? With the right training and the right processes, that's an entirely, it's not a biology issue. It's a totally process driven issue that should have been avoided. Wow, that makes you pause and think for a bit, doesn't it?
yeah, sure does.
Yeah, yeah. Splice started out as a clinical, most as you said, serving, hospitals and so, but it seems like, as you mentioned, you started pivoting a little bit to preclinical services. And I'll be honest, when the Biomed team, met you guys, and I can tell you, it's tough to it's tough to find and stay with providers in the preclinical space who hit all the hit all the check boxes: consistent quality, expertise in across different tissues, consistent pricing, consistent timelines. You're absolutely right. It's not just clinical that has like blown out timelines, sometimes preclinical as well. And the problem is when you're dealing with animal models, right? The clock is ticking. Because sometimes, you've got to make these decisions really quick to figure out how am I terminating my study? Am I going on with my study? So I think the clock ticks no matter whether you're in preclinical and clinical. So, but coming back to my question, what was it just kind of like happenstance and customer interest where you're like, oh, we'll try preclinical and go into non-human species?
The phones kept ringing. People were reaching out to us.
Oh boy, okay.
It is part of the DNA of Splice is that we're amazingly human. I'm typically involved with these customer phone calls, and I'm in the laboratory quite a bit. And so we started a veterinary client, so it got us into non-clinical, and then we realized we needed to bring in somebody that speaks this research language that could be a
Hmm.
consultative type of addition to Splice. And that's, I think you met Bruce, to really work with these researchers and
Yeah,
nail down that project,
yeah,
and do it quickly.
yeah. And it's very true because, especially when you're in the preclinical discovery stage, right? It's very open-ended because, I mean, I'm trying to draw a contrast to clinical here. But clinical, okay, this is my tissue. I'm staining for this marker, or I'm doing this, and it's a kind of a defined workflow. But a lot of times in preclinical, it can be very open-ended, and you have to go where the data takes you. Where if you have an unexpected finding, you're like, ooh, I was expecting to find high levels of X protein, and turns out I got Y protein or something. Another marker showing up. You have to pivot on a dime, which can be a little, unnerving and things like that. And the other thing I wanted to ask you is, with human, especially with IHC space, you have lots of antibodies available, commercially available, highly validated. But when you start going into non-human species like, maybe mice, rats, or even primates, sometimes it can be a little tricky on the reagent front. So has that been an issue for Splice? Does that become the rate limiting step, I guess?
We've got great automation with
Yeah.
immunohistochemistry, immunofluorescence, fluorescence, situ hybridization. But we have an instrument that also allows IHC development, and we do see that
Okay.
this does not mirror, our clinical IHC offering.
Right,
And
right, right. And yeah, tell me a little bit more about the automation because I have to say, when I toured your lab, I was like, these are some really cool instruments. And that's an important aspect of it because automation does reduce human error, especially for repeated tasks, and it also helps speed up things. So tell me a little bit more about what you guys have been doing as your demand has scaled, for things like auto stainers or liquid handling. So, what's been your favorite toys to implement?
there's a lot. So
Yeah.
um, we have a lot of customers that are in the same, phase of their company. They're startups, or they're raising money, and they don't have a lot of money to spend on capital.
Right.
So we want to bring in that equipment that they would have. They don't need to pay $200,000 or $300,000 for. And so the first one that was really fun was bright field scanning. So we brought in
Mhm.
automated Pramana, um, digital scanner, scans up to 40x. And,
Yeah.
we don't use that like other laboratories where it's integrated into your limb system, because we have now three clients, and we push that data into three different places, three different image management systems.
Okay.
So it's what's
Client specified image management. So each client is like, hey, I want my images to be in my system of choice. So they're very different. Is that what you mean?
exactly. We want to
Okay.
they want to be able to use that data in a system that they feel comfortable with. So we need to
Mm.
be able to push it there. So today we're doing Lumea, we're doing Proscia, and then we can just simply download them onto a hard drive and send them where they need to go.
Nice.
Um,
Nice.
so the digital scanner was a fun project, and right away we started getting like over 1,000 blocks a day. So we had to get an
Wow.
automated IHC or a H&E stainer. So we got the Prisma, Secure Prisma, and now we have two of them. One's for clinical, one's for non-clinical. And then we got a BioCare Encore Pro IHC. So the reason we like this platform is it's got several methodologies: IHC, IF, fluorescent in situ, all in the same instrument. And
Wow. That's
when
amazing.
you load reagents, um, you can get more out of these. Are all expensive, especially when you're doing multiplex spatial biology.
Mm,
So like we would charge a hospital maybe $10 for a block. We would charge $1,000 for one slide of a multiplex. Uh,
right.
but the reagents are hundreds and hundreds of dollars. So this instrument
Yes.
to get a little bit more out of it. And it's not, you can run different protocols. There's over 30 slides at a time. It's about a 4-hour run.
Yeah.
So we can kind of play and choose. And we do a lot of IHC development on that instrument as well.
Okay. So to pull on that thread a little bit more, because especially in the preclinical space, right? People are identifying new biomarkers, whether it's in cancer tissue or normal tissue or whatever, and they have their favorite marker. And you do get antibodies for, good number of these markers, but the IHC validation can be kind of, iffy or really not existent or, not done on the right tissue or something like that. So if we had, a preclinical discovery client saying, "Hey, this is my really uber cool biomarker, but I don't have an IHC assay and I really need one." So this is something Splice could handle, is if we got all the biology, the information about the marker and could buy a few, say, antibodies or, a few monoclonal clones or something, and you could develop the protocol. Is that something, is that accurate?
This is the importance of that consultative approach that we take. It's not up.
Right.
So we will get in. Some clients want to try their own antibodies, or they want us to get them.
Yeah.
I'll say a really important piece of this particular area is the control tissue.
Yes.
We've got a few other partners that have access to tens of thousands of tissue blocks with pathology data.
Oh,
So
that's nice.
you
Yeah.
understand, what you're trying to identify and find the right
Yeah.
control tissue. It plays
Yeah.
a role in developing that assay.
Yes. You're absolutely right. I didn't think of the control tissue, but that's absolutely right, because you've got to make sure that the signal you're seeing is actual signal and not some artifact or something like a little funky out there. Interesting. And one thing I remember when I noticed in your lab is, of course, you have all these you have excellent automation, but I was thinking a little bit about the amazing people that you have, because you have recruited your team of folks who handle these tissues. These guys are experienced, top-notch people. And I know that you've got a big, like, pool of people you can call upon, as needed. So tell me a little bit more about your staffing.
We've got a core team. There's 5 of
Yep.
us, and
Yep.
my laboratory manager. I've never seen someone who understands the histology process. So she can look at a slide and tell you, what happened with fixation or over processing or
Mhm.
under processing.
Wow.
Knows her way, and she trains all of us.
Yeah.
Troubleshooting machines. So the other folks have been I've worked with them in the past. We've put 2 students through the program that we have with the University of North Dakota. And I think you got to call out the resilience of this team. We work 24/7 when needed. So we have
Right.
a new customer in Pennsylvania sending a couple hundred blocks to us tonight. So
Wow.
the team will be in tomorrow. It's Saturday, but that's the life of a startup company. We're trying to keep expenses
Wow.
down. So you need just resilient people who believe in the company, and just kind of work around the clock until we get to the point where it's pretty stable and predictable volume.
And that's something that I think, really deserves a moment of, applause and just given its due, because that's part of it, right? When you're starting a new company or when you're doing something which has a ticking clock, and, histopath is a ticking clock where you need to get the data done in a certain time frame. I think the fact that you've assembled such a fantastic team, and it was my pleasure to meet some of them, it's yeah, I think that really does set apart Splice in many ways, because yeah, there's a lot of IHC and histo companies out there, but what's really struck me was the can-do attitude. And that's something, and it's not without compromise. We'll get this done in a time frame without ever sacrificing even a millimeter of quality or something. And that's something I think that's really noteworthy. So yeah, good job on building a fabulous team. It's really impressive.
Thank you.
Yeah.
Yeah, one other comment I'd say just in this topic, we do a lot of hospital-based work on, for
Yeah.
patients. When you think when a hospital's behind, a couple of weeks is typically the tipping point. What type of tissue are they okay parting ways with? And perhaps,
Right.
flying it halfway across the country for splice to
Yeah.
make beautiful H&Es. So it's your resections, it's your autopsies,
Yes.
that resections are kind of a treatment. It's not waiting for a
Yeah.
diagnosis. So you're a small link in the chain clinically.
Right.
So
Right.
excited with research.
Yeah.
Researchers, their whole life for this one opportunity to partner with Biomir, and get the animals and get them dosed. And histology is their endpoint. That's what's
Yes.
really incredible about this growth in our CRO business.
Yeah.
If we look at it, it is so important to
Yeah.
push their project to the next level.
You're absolutely right. And, I'll even add more on that because this is what we're seeing is because when clients come to us, to Biomir and Splice through our partnership, a lot of times they're coming to us, they've developed a brand new, drug or a class of a group of, candidates, and they need to make a go no go decision because a lot of times, for example, if they do a histopath analysis and they see crazy lesions or they see massive, cell damage or whatever it may be, it's they need to have that information to make go no go. And in drug development, especially preclinically, time is money, and everybody wants to be first, right? And even though it's a little bit more removed, from the patient, at the end of the day, everybody's developing these therapies because there are patients waiting,
Yep.
and they're on the clock too. So it's like there's this desperate need to basically for so many different disease areas. We've got to get moving faster. We've got to get the highest quality drugs into humans, into phase one clinical trials and so on. So yeah, no, that's absolutely, that's absolutely critical. And along that line, with any platform, analysis platform, data analysis is critical, right? Because you look at the picture, as you said, your lab manager can look at it and really give you some information. But for an average scientist, you're like, oh, okay. So what does this mean? Because, when pathologists read slides, it's like a encyclopedia for them. And the average person, maybe not so much. So, what's Splice's approach to analyzing this kind of data, especially in the preclinical space or non-human space? And what do clients get, I guess, once they've, at the end of their analysis?
So this is where partnerships are a key for Splice.
Yeah.
So we are, we're creating the data. We're never going to be an AI company,
Yeah.
but you got to make a beautiful H&E, and you got
Yes.
you got to scan it. So
Yes.
I'll go back to, without a beautiful looking H&E, the scanning is irrelevant. So, we
Yes.
focus on that. We focus on bright field scanning, thermal scanning, and then pushing. We want to push that data into a
Yes.
partner. And on the research side, there's so many players in this AI space to help researchers analyze their data.
Yes.
What's really
But as you said, I just want to interject, as you said, the analysis, whether it's AI or manual or AI aided or whatever, is only going to be as good as your starting image. Your starting image is fuzzy or unclear. You'll never really be able to get a meaningful data and a meaningful piece of data out of it, right? And I think that's an important thing because people get carried away from the data analysis, and sometimes people overlook the need to have, as you said, H&E, considered simple, but I think if you don't do it right, you're going to basically have nothing, right?
You got to make a beautiful H&E. And that starts the fixation, um, processing, the proper embedding, but even making a slide and cover slipping it. There's glue, and if you have a glass cover slip, there's air bubbles that could happen. So
Yes. Yeah.
QC throughout your whole entire process is critical.
Yeah.
We do have automated QC on our scanners that can look at an image around several different parameters for focus, for tissue folds. Um, so, right, but you need to have a beautiful looking slide
Yeah.
that gets scanned. Yeah.
And we talked about H&E, but I'm also, because one of the favorite, I think people look at IHC as well, either singleplex, your standard basic colorimetric IHC or IF or singleplex or multiplex IF. And that should increase the complexity of the data analysis, right? I mean, it gets a little more, or does it? Or
Absolutely.
what's,
Well,
yeah,
when you're standing for IHC, you can use bright field scanning, but
yeah,
if you have a multiplex, and people are talking about developing 100-plex, so imagine,
yes,
we've done a 6-plex PDL1 panel. So we
nice,
labeled that 6 different images on one slide. Where that's important is when you have limited tissue.
right,
But it's really expensive. So that's the other part. You could charge upwards of $1,000 a slide. And
correct,
when you scan multiplex, they're fluorescent images. So you need a different type of scanner that's
right.
really, really expensive. We don't get enough of that work yet. So we actually have a local partner that
Okay,
we go down and we use their scanner and they send histology work to us.
yeah.
So we get enough.
Which is entirely doable, right? Because once your slide is ready and cover slipped, it's very transportable.
Sure. I think with the fluorescent labels, you want to keep the slides out of the light.
Yeah, yeah. To avoid bleaching. Yeah. But you can just pop them in a box, seal them up nice and tight so that they're dark, and then basically take them and scan them anywhere. It's basically when the tissue is fresh that it's most fragile and needs to be processed. So there's a certain robustness that, for the actual slide and all the stain after the staining's complete.
Yeah.
Yeah. Excellent, excellent. And one aspect I wanted to touch on is the pathology analysis, and we've talked a little bit about this, right? You've got yeah. So, what's Splice's approach to working with pathologists to read the slides, in the clinical setting and the preclinical setting?
So on the clinical side, we're CAP accredited laboratory, so we
Yeah.
have a medical director, and he reviews our quality, H&E images, quality control. But we don't offer clinical testing
Okay. Yeah.
on patients. So Splice defined as creating an H&E, it's actually not a test. CLIA doesn't consider it a test.
Oh, okay.
We're making a slide. So there's
Right.
no testing for them to come out and inspect. But on the research side, we brought on a veterinary pathologist who works out of BU, works for us on a per diem basis for certain projects. And we bring him in to the initial discussions on, working with our customers, developing a statement of work. And as needed, client, he'll jump in and do vet path work, and has a lot of
That's
experience
awesome.
with, small animal studies.
And that's really good to hear because that's, something from our clients' perspectives because people who outsource IHC or, these kind of studies typically don't have access to a vet path, or if they do, it's probably not, very frequent. So I think having that component of having, Biomere doing the dosing, the in-life studies, and then collecting the tissues, and then kind of handing it off to you guys to work your magic, generate gorgeous images, like super, clean data. And then I think the final piece of that puzzle is having that data analyzed by a vet pathologist, as you mentioned, to generate a report saying these were the findings, because I think that's the complete workflow. It's like I see that three big kind of chunk segments or steps. And I think, it works for both sides because we do what we do. Like Biomere knows how to do in vivo. You guys are clearly experts in the histo area, and you have an expert pathologist to kind of close out the loop and round out the workflow. So I think that's kind of a really interesting aspect. So I wanted to ask you, as we start wrapping up, what are your plans for Splice? Because I know you guys are in a really, really nice space, but it seems like the demand is only going to grow. So what are your plans for growing Splice?
So we began investing in contract research in
Yeah.
terms of people and capital and equipment last summer. And
Okay.
um, what we saw was, I think, a little bit of uncertainty in the market towards the end of
Mm.
last year as we
Mm.
were ramping up our marketing. The holidays were coming. So we spent the last 6 months really just getting the word of Splice out there
Yeah.
and visiting places, going to local meetups. Um, and finally, in January, really started to see growth in our contract research.
Yes.
So as a percentage of our business, I'd like to see research about 50%. Um,
Okay.
it's not there quite yet, but it's about 20%,
Yeah.
25%. But this space, it's there's so much demand, uh,
Yes.
and given our geography,
Yes.
this is where a lot of it happens. We have
Yeah.
local couriers. We people can come and see us. We can come and see them.
Yeah.
So on the hospital side, what we're trying to do is be less of a backup situation and find customers that have a real consistent need.
Yeah.
And we actually just sold our first subscription model to one of the
Oh,
largest
congratulations. That's very cool.
one of the largest health systems in the country, for over 40 hospitals. So they subscribe to Splice. All of their locations have access. They have the ability to stat cases digitally. But these are places that they're really large, 2,000, 3,000 blocks per day. And they're always going to have this need. So why not
Yeah.
subscribe to Splice?
Yeah.
I think getting a consistent, predictable revenue is top priority. Growing our subscription, and then research. It's turning into, like, let's just keep up with it. There's a
Yeah.
lot of work that you see
Yeah.
when you sit down
Yeah.
with a customer. Research, it's a process to develop a very thorough working document of what the goal should look like. Um, so yeah, it's growing the contract research work, um, converting from being a backup lab to hospitals to being a consistent
Love it.
part, normal part of their business. And then occasionally we got a call from an international veterinary
Mm.
out of Louisville, and they said, "Hey, I got to send you a couple thousand blocks. I'm behind. It's going to be a one-time type of thing." So you manage through
But
some.
word spreads, right? Like that's the other thing. It's word of mouth. For example, I think, when we heard about you guys, you know, I don't think we can overemphasize the importance of local, right? Because when we're dealing, again, we talk about the fragility of tissues, and tissues are more robust than others, but the comfort level that biomere scientists have going, hey, I just I know it's like less than half a mile away, so I know that my tissues are not sitting in fixative forever and ever and ever, and I can get these tissues, properly processed and ready for staining. That actually, in some cases, means the difference between no data or no stain and very, very informative staining data. And that's a big deal because, in vivo studies, there's a lot riding on these, and not just, from a setup and budget and timeline perspective, but also from the aspect of, you know, responsible animal use, right? And so you want to make sure when you do run in vivo studies, you're getting all the data you possibly can to make informed decisions. And I think partnerships like this is just another way of making sure that we're being very responsible about designing and executing our client studies.
Our
So,
geography is a huge benefit.
yes,
Um, in addition to Biome, we have several other clients that visit.
I love it.
They come here, they want to participate in their projects. We had one come in this morning, actually,
Yeah,
pick up slides. They're a local CRO, wound testing CRO,
yeah.
Framingham.
And that's the transparency aspect of it, right? The more transparent, people are, and they know where their tissues are, where their data is, what state. I think it just builds more and more client confidence, de-risks delays, or if there's issues, right? You're like, upfront, okay, this tissue is not looking quite right or was damaged, so we better like inform. So I think those are all the aspects that are so important. So yeah, I'm so glad. I'm so glad I can tell you the entire team at Biome is so excited to be partnering, and it's an absolute pleasure working with you and Bruce and Evan and knowing that you guys are right up the street from us. So yeah, and we're so excited to offer our clients, these expanded services in partnership with you guys. So with that, I will say thank you, Tom, for joining me and for sharing your vision and sharing so much great information on Splice's capabilities. What I'll say to our listeners is if you're interested in learning more about our services online, check out biomea.com, or you can email us at bd@biomea.com. And definitely check out Splice as well as Biomea's LinkedIn pages. We're always posting new information. We're so proud to have announced this partnership in January, just I think last week. And we're looking forward to a really exciting collaboration, not just for client projects, but also for some really interesting internal research projects, which I hope can be presented in the forms of posters and papers. So with that, Tom, I will say thank you, and looking forward to a great 2026 and a great collaboration.
Same here, Angelique. Thank you so much for having me.
Absolutely.